The central nervous system is well protected. The brain has the bones of the skull, the membranes or the meninges, the cerebrospinal fluid, and the blood-brain barrier. The meninges cover and protect the central nervous system. They help protect the blood vessels and enclose the venous sinuses. The cerebrospinal fluid circulates in the meninges. Also, the meninges form partitions in the brain. They're formed of connective tissue, and we have three layers. The Dura Mater is the outermost, the Arachnoid Mater is inner, and then the Pia Mater directly covers the brain. The Dura Mater is the toughest of the layers. Mater means mother and Dura means tough, so this is the tough mother. It is a double layer. There is a periosteal layer that's the outer layer. It's fused to the periosteum of the skull. The meningeal layer is the inner layer. It forms the dural sheath in the spinal cord. The layers are typically fused except at the dural sinuses. The dural sinuses are found in the longitudinal and transverse fissure, and they collect venous blood and direct it down the neck into the jugular veins. The meningeal layer also forms the dural septa. The dural septa help limit excessive movement of the brain. The falx cerebri is in the longitudinal fissure. It's attached to the crista galla. The falx cerebelli separates the hemispheres of the cerebellum. And the tentorium cerebelli is a horizontal dural fold. It covers the cerebellum, separating the cerebellum from the cerebrum. It's in the transverse fissure. This is an autopsy specimen, and the Dura Mater is exposed here. You can see it's a very tough, fibrous kind of membrane. Here's the brain under here. Here are the various dural folds. The falx cerebri in the longitudinal fissure. Here will be the superior sagittal sinus, the falx cerebri between the hemispheres of the cerebrum, and the tentorium cerebelli. And here will be the transverse dural sinus. In this autopsy specimen, again, you see the dura. Here is the superior sagittal sinus, the transverse sinus. Here is the brain that's been exposed with the dura removed. This would be the falx cerebelli between the cerebrum and the cerebellum, and the falx cerebri would be in this fold, the longitudinal fold. The arachnoid mater is the middle layer of the meninges. Because of its web-like extensions, it gets the name spider mother. It is separated from the dura mater by the subdural space, and then there is the subarachnoid space between the arachnoid mater and the pia mater. The cerebrospinal fluid circulates in the subarachnoid space, and the largest blood vessels of the brain are located here. There are arachnoid villi, or arachnoid granulations, that project into the superior sagittal sinus. This is one of those dural sinuses. This allows the cerebrospinal fluid to be reabsorbed back to the blood. The Pia Mater is a delicate membrane that clings directly to the surface of the brain. Pia means gentle, so this is the gentle mother. There are lots and lots of tiny blood vessels in the Pia Mater. If the meninges become inflamed, we have a condition known as meningitis. The inflammation may be the result of either viral or bacterial infection. We can diagnose meningitis by doing a spinal tap. The inflammation may spread to the brain, in which case we will have encephalitis. This means the brain tissue itself is inflamed. The protections of the brain include the skull bones and the cerebrospinal fluid, which will flow in this space. Here we see the dura mater, the periosteal layer, and this would be the meningeal layer, and that comes off to make the dural fold, the falx cerebri in this case. And this is one of the sinuses. This is the superior sagittal sinus that's in the longitudinal fissure. The arachnoid mater has the arachnoid granulations or arachnoid villi that project into the superior sagittal sinus, and these web-like extensions that attach it to the pia mater. The pia mater is directly clinging to the brain, going down into all of those sulci and covering the gyri. Here is the subdural space between the dura and the arachnoid, and here is the subarachnoid space. This is where the cerebrospinal fluid will flow. The cerebrospinal fluid acts as a liquid cushion for the brain. It also helps nourish the brain and provides chemical stability. It is a filtrative plasma, and we make about 500 milliliters a day. It differs from plasma in that there is no protein in CSF, and only about 2/3 of the glucose level that's in the blood gets to the CSF. There is more vitamin C in the cerebrospinal fluid than we find in the blood. Sodium, potassium, and the other ions are present in both the CSF and the plasma, but they're in different ion concentrations. At any given time, there's a total CSF volume of about 150 milliliters, and we replace that about every 8 hours. Cerebrospinal fluid is formed by the choroid plexus in the roof of each ventricle. The choroid plexus is a cluster of capillaries that's covered with ependymal cells. The ependymal cells provide a barrier between the blood and the cerebrospinal fluid. There are tight junctions between the ependymal cells, and this helps control what leaves the plasma and gets into the cerebrospinal fluid. The cerebrospinal fluid will move through the ventricles and down the central canal. Most of the cerebrospinal fluid can leave the fourth ventricle to get into the subarachnoid space so it can circulate around the brain and spinal cord. It is reabsorbed through the arachnoid villi to the blood in the superior sagittal sinus. The Pia Mater will have those tiny capillaries in it, and as the blood flows through these capillaries, we will force some fluid out into the cerebrospinal space. And as the blood flows through here, we will force some of the fluid into the cavity of the ventricle. The ependymal cells, and these are ciliated to help improve the flow, have tight junctions, so things have to diffuse through them to get into the cerebrospinal fluid. This will slow down the movement of many things, so less gets into the cerebrospinal fluid, and some things won't get in at all. Once the fluid is formed, it will flow through the ventricles and down the central canal. Most of it can leave the fourth ventricle to get into the subarachnoid space to circulate around the brain and around the spinal cord. As it comes back up to the top of the head, the arachnoid villi will project into the superior sagittal sinus. There's very low blood pressure in this space, so the pressure behind the CSF forces the fluid back into the blood. So it's filtered from the blood, circulates around the brain and spinal cord, and returns back to the blood. We're constantly making and reabsorbing cerebrospinal fluid. Hydrocephalus occurs when there is an accumulation of cerebrospinal fluid. This can either be because of an overproduction or because there's a blockage of flow or reabsorption. In infants, the skull bones are not fused, so as the fluid builds up, the head will enlarge. In adults, the bones are fused. The buildup of fluid puts pressure on the brain. It causes headaches and other central nervous system disturbances. We can treat this by draining off the excess fluid. A ventricular shunt can be put in that will take the fluid from the brain down to the abdominal cavity, to the peritoneal space. Here is a picture of an infant with hydrocephalus. You can see how the pressure has enlarged the head. Not only will the head be enlarged, but when they look at images of the brain, the ventricles of the brain are also enlarged. The blood-brain barrier is the function of the astrocytes. These cling to the capillaries. Also in the brain, there are tight junctions between the cells of the capillaries. This helps control what leaves the blood and gets to the brain tissue. Glucose, some amino acids, and electrolytes pass fairly easily, but metabolic waste, potassium, proteins, and many toxins can't pass. The blood-brain barrier is ineffective against anything that's fat-soluble. So fats, fatty acids, oxygen, carbon dioxide, and any other fat-soluble molecules can get through the blood-brain barrier. This includes alcohol, nicotine, and anesthetics. Some areas of the brain don't have a blood-brain barrier because they need to be able to sample the blood. The vomiting center, for example, needs to be able to identify blood poisons, and the hypothalamus has to be able to look at all of the blood chemical levels. This is the major homeostatic center of the body. The blood-brain barrier is not fully developed in the newborn. Traumatic brain injury is the leading cause of accidental death. The coup injury is the localized injury at the site of the blow to the head. The contra-coup injury occurs if the head is hit hard enough that the brain actually ricochets off the other side of the skull. Then we will have an injury on the opposite side of the brain. That's the contra-coup injury. Concussions are slight injuries. They're fairly short-lived and there is no permanent damage to the brain. A contusion is basically a bruise. Here there's going to be some destruction of brain tissue. The individual can remain conscious if it's a cortical contusion, but if the brainstem is contused, then you can go into a coma. Subdural and subarachnoid hemorrhage is when we have intracranial bleeding. The bleeding is either into the subdural space or the subarachnoid space. As the blood comes in and clots, it causes pressure on a part of the brain. We treat this by removing the hematoma and repairing the area. Cerebral edema occurs if the brain is damaged and swells, becomes edematous. We have to treat this with anti-inflammatory drugs to reduce the swelling. Cerebrovascular accidents, or CVAs, are also known as strokes. This is the most common nervous system disorder and the third leading cause of death. In a CVA, blood circulation to the brain is blocked. That leads to ischemia, damage to the tissue as a result of low oxygen levels. Clots are the most common cause, but hemorrhage and edema may also be a cause. Because the brain is set up to be contralateral, if the stroke is on the right side of the brain, the left side of the body will show the effects. TPA, or tissue plasminogen activator, is the only approved treatment for CVAs. This is a clot buster. It will dissolve clots. Transient ischemic attacks, or TIAs, involve short loss of function, 5 to 50 minutes. There's numbness, impaired speech, or paralysis. These are reversible and function returns fully. Sometimes these are a red flag for stroke. About 30% of people who experience TIAs will go on to have strokes within the next 5 years. Some physicians prescribe aspirin prophylactically to prevent the development of clots. There are a number of degenerative brain disorders. Alzheimer's is one of them. This is a progressive degeneration that leads to dementia. 5 to 15% of the people over 65 have Alzheimer's disease. There are multiple forms of this disease, and some of them are genetic in nature. With Alzheimer's, there's memory loss, short attention span, and disorientation. Eventually, they lose language, they become irritable, moody, confused, and may even experience hallucinations. Acetylcholine seems to be a neurotransmitter that's involved in memory storage and retrieval, and in Alzheimer's, there seems to be an acetylcholine shortage. Most of the drugs currently given to treat Alzheimer's are aimed at building up acetylcholine levels. In the brains of people with Alzheimer's, we see plaques of beta amyloid peptide that form in the brain. This may involve an infection with a prion. This causes the production of abnormal proteins. Prions were involved in mad cow disease. As a result of these plaques, we see neurofibrillary tangles developing. The neurons here die. The brain shrinks at a higher rate than normal. Parkinson's is another degenerative brain disorder. Here there's a degeneration of dopamine-releasing neurons. The basal nuclei are deprived of dopamine and they become overactive. This causes tremors, a forward-bent posture, shuffling gait, stiff facial expressions, and slow movement. Remember, the basal nuclei were involved in smooth movements and controlling extraneous movements, as well as starting and stopping movements. While the cause is unknown, it is seen in older people in their 50s and 60s. This may be one of the diseases that's linked to mitochondrial DNA, or there could be a protein degradation problem. Some drugs help alleviate symptoms but don't cure it. L-DOPA will transform into dopamine so that we can have a little more dopamine in the system. Deprenyl will slow the deterioration of the basal nuclei. Research is currently going on looking at intrabrain transplants of embryonic tissue to replace the lost neurons. Huntington's is a hereditary dominant disorder. Symptom onset occurs in the mid-30s, and usually the disease is fatal within 15 years of symptom onset. Huntingtin protein accumulates in the brain. This causes neuron death and leads to massive degeneration of basal nuclei and the cerebral cortex. The initial symptoms are wild, jerky, flapping kind of movements of the arms. Chorea. Later, there's marked mental deterioration. In this case, we have too much dopamine, so these people are given drugs that help block dopamine effects, dopamine blockers. There are a number of diagnostic procedures that can be used to help diagnose central nervous system disorders. Reflex tests can assess neural function. They can detect intracranial hemorrhage, multiple sclerosis, or hydrocephalus. Pneumoencephalography is a special X-ray of the ventricles. It can be used to diagnose hydrocephalus. A cerebral angiogram assesses the arteries of the brain. A dye is injected into the arteries of the brain, and then an X-ray is taken to see if blood is flowing as it should. MRIs and CTs help detect brain lesions, tumors, multiple sclerosis plaques, and areas of dead tissue. And PET scans can help find seizure lesions and help diagnose Alzheimer's.